Okay, so we're recording. So let's get started. Unless somebody has any questions right off the bat before we get started. All right, I'm going to share a screen with you and just point out what you need to know for the reproductive anatomy lab quiz next week. So let me look my, oh wait, share screen with the Mayo lab right here.
I don't see the lab packet for that.
Oh, here, I'm sharing my, you don't have this document?
No, I don't see it in D2L in lab or lecture.
Well, let me go in there and tell you where it is.
Oh, never mind, found it, sorry.
You got it? Okay. Yeah, it has a male lab and female lab and reproductive lab.
Yeah,
That's
I was just looking for one big thing.
Okay. All right. So yeah, so it's like 3 documents. First on male anatomy, female anatomy, and then development lab, and that's only one. Okay, so there's so we pull up the pictures of the male reproductive model, male pelvic model, model of so it's actually there's two male and female models. One's a sagittal section where you can't see everything, and one's like 3D. All you can see all parts of it. Unfortunately, the picture of the female 3D, I call it 3D, is not in the female lab. We should have updated it, but I'm going to show you a picture of what it looks like. I found a picture in on the web, and I'll show you what it looks like. A model of developing embryo. This is the so that's really mostly the next chapter, chapter 29. So this is going to be both chapters, the reproductive anatomy and then development, which is the very last chapter. One model for that. I'm not going to do any slides, and the preserved animal specimens is just going to show you a couple things in the animal specimens. So so here we go to the left here on a model of male reproductive system. Just about that's really it for the. So I'll go through each one of those, and that's it for the male. There's less on the male than a female. So I'll go through all of these, starting with the sagittal section model here to the right. And so this is the when we have in. So you got to know the all the parts of the male urethra. Remember, there's 3 parts to the male urethra: the prostatic urethra that goes to the prostate gland, the membranous urethra, which is right here. It's very short, and it goes through what's called the urogenital diaphragm, and then the spongy urethra. It's called a spongy because it goes through erectile tissue called the corpus spongiosum in red here. And then it expands at the head of the penis to the glans penis, which is the expansion of the corpus spongiosum. And the glans penis, if you're not circumcised, covered by a prepuce or foreskin right here. And there's blue right here, and there's 2 of these. They are also erectile tissue called the corpus cavernosum. Then there's going to be some. So this is the urethra. Now, right inside the prostate is a little another tube called the the ejaculatory duct, which is the really end of the ductus deferens or vas deferens. And you can see that better in the 3D model, which I'm going to show you in a moment. There's another little gland right here called the bulbourethral gland right down here that really makes a pre-ejaculatory fluid. But the semen, of course, is composed of secretions from the prostate and the seminal vesicles, which you can't see in this sagittal section because they're over here behind the prostate on both sides of the vas deferens. So we'll see that in the next model. You don't have to worry about urinary bladder, pubic symphysis. I'm not going to ask you. That's for rectum. We'll pass that. Okay. This model here, let me get to the next page. Right here is the newer model, which I call the 3D model. And it's a better model, as you can see everything. And because remember on this model over here, you can't really see a testis or a spermatic cord or anything because it's in cross section. But here you can see testes, two testes. Well, this one's more dissected on this side than on this side. And there's a testis right there, what's called an epididymis right here, which is a storage area for sperm. And this obviously is the scrotum, and the scrotum is covered by a muscle called the dartos muscle. These are spermatic cords here that the testis is at the end of, and they go through a space in the male abdominal wall called the inguinal canal. And they're full of a series of blood vessels called the pampiniform plexus. And it's going to be actually a countercurrent mechanism to keep the testes cool. That's, okay, I mentioned this in the lecture, why do we need a scrotum? We need the testes have to be held outside the body because they have to be 2 degrees below body temperature. If they're not, a man makes all kinds of deformed sperm. Spermatogenesis doesn't work well at body temperature. It has to be 2 degrees below. So that's why we need a scrotum. And it's kept cool by being outside the body, but it's also kept cool because of this countercurrent mechanism in the pampiniform plexus. We have arteries coming down to the testis bringing hot blood, and you got veins going back into the body carrying cooler blood, and the heat from the arteries goes into the veins, and the heat goes back in the body rather than going out. So the blood that goes to the testis is actually a little cooler. Also, this cremaster muscle right here. This is also a temperature regulation device. It's I guess it's skeletal muscle, but it's not controlled. But it's controlled by the autonomic nervous system, and it will pull the testes up closer to a man's body if it's warm out. And they'll relax and let the testes hang lower, farther away from the body if it's warmer out. Okay, now, and in the spermatic cord is the ductus deferens, also known as the vas deferens, VAS, or ductus deferens. It's the sperm duct, right? And so if you look on the back side, you can see it better. Right here is the back side of that same model. And here's the ductus, that's a ureter. So the vas deferens or ductus deferens hangs right over the ureter and heads toward the prostate. But here is that other male gland I was talking about, seminal vesicle on both sides, and they so the vas deferens goes right through there, and those uh, glands contribute fluid to semen. Okay, and then we go on cross section here. We can see the other stuff that was on the cross sectional model, the same things, the two erectile tissues, the parts of the male urethra, the ejaculatory duct, and so on. Okay? That clear? Everybody got, if you got any questions, and here you're all laid out right here, all the stuff you need to know. So if you got any questions, interrupt me anytime you got a question. I might do something with the cat. Usually, only I do with the cat is I dissect out a testis and show what see what a scrotum and what a testis is, and look at the ductus deferens, because you can see how to come around and go behind the bladder and into the seminal vesicles, although I never dissect out the seminal vesicles or the prostate. So it's really just a ductus deferens or a scrotum or a testis in the male cat. So I don't dissect out the penis or anything else. And here's some slides. I'm not gonna, like I said, I'm not gonna do any slides. So, oh, and not a slide of sperm either, but there is a sperm model. It's on the actually, it's on the developmental panel. It's this big flat model that has all different stages of well, fertilization and the beginning of the cleavage divisions and everything. It also has a sperm on this panel, and that's the sperm. And so you need to know parts of a sperm. And here's the head of the sperm with the 23 single chromosomes, head with acrosome. Now, an acrosome is a very tip of the sperm head. It's got digestive enzymes in it, and it breaks open in what's called the acrosomal reaction. I talk about that in the lecture. That's to break away the outer cells around the ovulated object and get to the membranes of the digest to the membranes of the oocytes where fertilization can occur. They don't really get. They should mark a line at the top here to show you where the acrosome is, but they don't. Then you get the mid piece right here. This is filled with mitochondria that break down sugar, which happens to be in semen, this fructose in semen, to fuel this flagellum right here, to make it mobile and have it move from the vagina up to the fallopian tubes for possible fertilization. Okay, so you need to know the parts of the sperm, and I'm not going to do cross section of penis or thing. Now, the lab report, I'm just going to not label all this stuff or put this, but give you the right numbers for this page 14 here. So I went through and one of them I really don't understand, but I'll give them to you. So here on the left, 14. So the very first one is 14, testis. Second one is 7 epididymis. Third is 10 prepuce. Here's the one I don't understand: extension of internal iliac. I don't know what they're talking about there. I had to use process of elimination for me to figure this out, and the process of elimination says 4, the cremaster muscle. But extension of internal iliac, that's a blood vessel in your leg. So I don't know what they're talking about. This might be a typo. Anyway, so the cremaster muscle, remember, is this thing over here that lowers or raises the testis depending upon the temperature. Okay, that's a strange one. Okay, after that is 5, ductus deferens, 2 weak sense from epididymis to ejaculatory duct. Next is corpus cavernosa, 2 cylinders of erectile tissue, which is 3. Next one is 11, prostate, gland surrounds urethra, inferior to bladder. The next one is 2 corpus spongiosum, cylinder of erectile tissue surrounding the urethra in the penis. Next one is 8, inguinal canal, opening the abdominal wall. Next one is 12, seminal vesicles, 2 glands posterior to urinary bladder. Next one would be 6, ejaculatory duct, small tooth and vas deferens to urethra. Next will be 9, membranous urethra, passes through urogenital diaphragm. That's the shortest part of the male urethra. 2 small glands at base of penis. It's going to be 1, the bulbourethral glands. And the last is going to be 13, cord of connective tissue containing vas deferens, testicular arteries, pempiniform plexus, which contains the testicular arteries. I don't know why they put that twice. Lymphatic vessels, well, we didn't mention those, and nerves.
Dr. Murphy,
Yes.
do you mind just going down that list again and just giving us the numbers so I make sure I matched them right?
Sure, sure. Okay. From top to bottom, 14, 7, 10, 4, which is the one I think is bizarre, 5, 3, 11, 2, 8, 12, 6, 9, 1, 13.
Thank you.
Yeah? All right, good. And I think that's it for the male. Unless anybody have any questions about the male structures. Okay, we'll go on. Wait a minute, let me close that. And go on to female. Okay, there's going to be more things in the female. First, the models of female reproductive system. Most of these portions can be done on the sagittal section. It's not really anything hidden. Like in the male, there's stuff hidden. You can't see it. But you can see just about everything on the sagittal section, all of these things right here. And in preserved animal, you can see ovaries, uterus. I usually don't try to find a vagina. Usually look at just the uterus and the ovaries. And the uterus in cats, it looks really different than human uterus. It's a big Y-shaped thing with uterine horns because cats have litters, and each fetus is going to be lined along these horns. You know, human uterus is sort of like triangular shaped, and it's called a simplex, versus these uterus with uterine horns like in cats. Okay, model of ovary. We're gonna have a big model of the ovary, and it's gonna look mostly this is going to be the developing secondary oocyte that's going to be ovulated, different stages of it, follicles, follicular development. Okay. And so this is the model of ovary. A prepared slide, I'm not going to do the slides again. We got a breast model. I don't really consider a breast a reproductive structure. It's more like important for the products of reproduction, but we do have a breast model. And there's only a couple things on that. And then, so here is the cross-sectional of the female. And starting with the, well, let's go with the uterus right here. There's a bunch of parts of the uterus. Like the stomach, it has certain parts. This is the fundus up here. There's nobody in the waiting room, is there? Wait a minute, hang on. Oh, no, okay. So, matter of fact, it's pretty similar to the stomach. It has a fundus up here, a body right here. And then the cervix is the most inferior part right here, the cervix. And it's the tube through the cervix is called the isthmus. And but you got to also be careful about the layers, because this is could be the body, but it's also the myometrium, which is the muscular part of the. So if that was on a multiple choice, you know, you could figure out which I'm referring to, but because both body and myometrium wouldn't be on the same list of choices, right? Because arrow pointing there would be, it could be either body or myometrium. And then the dark area on the inside is the endometrium, the lining of the uterus. I don't really use the term parametrium ever, the very outside. And I don't use recto-uterine pouch either. I never asked that. Now, here's the ovary right here. Now, there's your uterine tube. Now, that's also got another name called the fallopian tube. I usually use fallopian tube. But uterine tube is fine also. But if it was, I was going to ask that on a multiple choice question, I'd probably put fallopian. Okay, you don't have to worry about the peritoneum. You have to about the female urethra right there, vagina right here, and the clitoris right here. Oh yeah, there's one thing you really can't see on the outside. That here's the labia minora right here. Labia majora will be on the very outside, and it doesn't show well on this model. You have to look at the 3D model. And I'm going to show you a picture from the web of the 3D model. Now, one thing is unusual are these ligaments. You got to learn these ligaments, and there's several of them. With the uterine tube or fallopian tube, I'm not going to be asking about these specific areas. The ampulla, the infundibulum. This is where the fimbria are that surround the ovary, and the isthmus here. Now here are these ligaments, and they've got some strange name, mesosalpinx. So that's between the, I guess to remember where these are located. Between the fallopian tube and the ovaries are mesosalpinx. The ovary is attached to the body wall by what's called the ovarian ligament. Then on the other side of the ovary, the inner side, is the broad ligament. And then finally over here, this cord-like ligament is called the uterosacral ligament. And I think it holds the uterus in place. And obviously, ovarian ligament holds the ovary in place, and the broad ligament on one side, the inside of the ovary, and the mesosalpinx on the outer side of the ovary. All right. Let me wait a minute. And there's all the structures, anatomical structures. The female right there. And yeah, so you don't have labia majora here because you really can't see it. So I'm going to pull up the, see, where is that? Oh, wait a minute. It's, where is that? Ah, female anatomy model, right here. Uh-oh. What's wrong? Oh man, it's not coming up. All right, hang on a second. I'm gonna go, let's kill this. Okay, I'm gonna go on, let's stop sharing for a moment, go onto the web and find that model again. Let's see. So if I can find it again. I'm not screen sharing because I have to go into there's always a danger going into the web looking for reproductive stuff because you might go to the wrong place. You know, and some of these pictures might not be appropriate to be sharing with you. I got to be careful. Where is, oh, I don't see it. Let's see, where is it? I brought it, I downloaded it, I put it on a Word document, and I thought it would. Okay, where is it? Where is it? Where is it? Come on. I think I've got, wait a minute, I think I've got a picture of it. Oh, where is it? Oh, there it is, right there. All right, let me close this stuff. Go back to screen share, and I should be able to here it is. Okay, this is the what I call a 3D model, female, right here. Unfortunately, we can't look at real close, but it's the good thing is you can see the labia majora right here, which you can't see on the cross section. But everything else you can see fortunately on the cross section, and this thing opens up also to get a cross section inside. So I'd be probably putting some questions on this particular model right here. So sorry, we should really update the female lab document to show photos of this particular model. It's a new model, and we just haven't put it up yet. Anyway, so is everybody clear?
Yes.
All right, let me stop sharing that, and then go back to the lab document. Right there. Okay. And there's the cat. Oh, let me make sure nobody's waiting. Oh, okay. And here you can see the uterine horns, these long, thin things. That's really different in cats than in people. And the ovaries are right up here and up here. So that's all you really need to know is the ovary and the uterine tubes for the female cat. Okay, now we'll look into the ovary model here. And there it is right there. And these are all different stages of follicular development. So it's a development of what's called secondary oocyte, which is going to be the egg cell that's going to be ovulated. And so you start off with primordial follicles over here. And these have an oocyte in them. And a woman is born with all of them she's ever going to have. And inside is a oogonium that has already started meiosis, is arrested in prophase 1. And over the course of the ovarian cycle, over the first half of the ovarian cycle, from actually starting in the menstrual flow of the last period to the 14th day of the present ovarian cycle, they develop. Some few of these are selected by some mysterious process to continue meiosis and develop into a what's called a primary oocyte that will be ovulated. So it goes from the primordial follicles to what are called primary follicles. Now this down here is in a different model. This is the, it has some of the same stuff. And also there's an error here that I have to point out. So this is one model, this is a different model, but it points out sort of like the same stuff. So these are primary follicles here. And this is, so here's a primordial primary follicle. And I think this is wrong, the secondary follicles. This is a primary, that's a primary, that's a primary. This is a secondary, and that is, it has an antrum, there's a space. So if you see a space, it's a secondary. If you don't see a space, it's a primary. And the cells around here are called the granulosa cells. And it eventually becomes what's called a mature or Graafian follicle. Graafian with two A's, Graafian, Graafian, or mature follicle right here. It's got a huge antrum. It's got a little peninsula of tissue here where the secondary oocyte is located. Now, this whole thing right here this is really bizarre. It doesn't have a name. It's blown up from right here. Right here is where it's located in the Graafian follicle, and here it's blown up to see the parts of it. And it is what is ovulated. I just call it the ovulated structure. There's no anatomical name for it, which is, I think, bizarre. Because what is ovulated is we think that, an egg is ovulated, but actually it's a multicellular thing that's ovulated. This thing right here is a secondary oocyte, and there's the nucleus. Then it has a clear coat around it called the zona pellucida. And then it has these cells, which used to be granulosa cells, surrounding the secondary oocyte, and that's called the corona radiata. Interesting, it's the same as the name has some white matter in the brain, corona radiata. Anyway, so, and then this little thing right here is the first polar body. So remember, this oocyte starts off arrested in prophase 1. As it develops the follicle, it goes through the rest of meiosis and becomes arrested here at metaphase 2. And then it's ovulated as metaphase 2. And so anaphase 1 has occurred, and you've separated homologous pairs of chromosomes. And you're only going to save one set of homologous chromosomes. You're going to discard the other set. And that's what the first polar body is. It's 23 perfectly good chromosomes just going to be thrown away. And they're replicated chromosomes. Okay, and this thing is going to last about 48 hours in a fallopian tube. And if it's not fertilized, it will disintegrate in 48 hours, after 48 hours. And if it's not fertilized, it will disintegrate still arrested in metaphase 2. It won't even complete meiosis if it's not fertilized, because what's the point? So you know that. And so over here also it's a mature follicle, and here ovulation is occurring right here. This is what this thing is about to pop out, and it has to be caught by the fimbriae surrounding the ovary, and so the cilia in the fimbriae bring it in, and the cilia bring it down the fallopian tube, where it will be fertilized if there's sperm present there. After the ovulated structure leaves, then the remaining cells here become this thing called the corpus luteum. And it is going to be a source of progesterone in the second half of the ovarian cycle. And it's going to prepare the endometrium for possible implantation. Right? The very outside of the ovary is called the tunica albuginea. These are blood vessels, mesovarium up here. And if there is no implantation of a blastocyst and the pregnancy doesn't start, then this corpus luteum will degenerate into this thing right here, or this thing here in cross section called the corpus albicans. And then the progesterone will drop off. the stratum functionalis of the endometrium cannot be supported without progesterone, starts to disintegrate, and that starts the menstrual flow. And then during that menstrual flow, some FSH is secreted, and more primordial follicles are selected to possibly become the egg of next month. Right. The ovulate structure of next month. Okay. I think that's everything there. Okay, so I'm not going to give you a slide of the ovary. I'm not going to give you those things in a slide. Or of the uterus. But the slide of the uterus is really distinctive. It has all these spiral arteries and everything. It's really distinctive. There's the breast model. We got a couple things. You got the nipple, the areola, there's lobes, and there's little lactiferous ducts here. Whoops, I'm pointing my finger. Lactiferous ducts, lactiferous sinuses, and some adipose tissue. Only a couple things in the breast model. I'll probably just have like one question on the breast model. Okay, so now I'm going to do the same thing here. With the male, there's 15 structures, and we're going to go right down starting with the top to bottom. Pear-shaped hollow organ is uterus, 14. Thin-walled fibromuscular tube is going to be 15, vagina. Third one is going to be 10, paired and have the shape of almonds, are going to be the ovaries. Next enlarged region of uterine tube is 1, the ampulla. Cord-like attachment of uterus to the sacrum is 12, the uterosacral ligament. And delicate hair-free skin folds of external genitalia is 7, the labia minora. Tubes that extend from the ovary to uterus, 13, the uterine tube or fallopian tube. Next one is 3, most inferior portion of uterus, cervix, 3. Inner lining of uterus, endometrium, 4. Finger-like projections of fallopian tubes are the fimbriae, 5. Anchors over from above to pelvic wall is 11, the suspensory. Now, wait a minute. That was I think it's going to be broad ligament. Change that to broad. There was no suspensory ligament on this model. I don't know where they got that. So make that the broad ligament right here. Okay. I don't know why they did that. Change the names. That's 11. Okay. Yeah, anchors over from above the pelvic wall. Modified pseudoriferous, modified Swick glands, 8 mammillary glands. Raised area of uterus, going to be 6, fundus. 3 layers of smooth muscle and wall of uterus, the myometrium, 9. Peritoneum that extends from the wait a minute, that's broad ligament up here. I'm sorry, the peritoneum that extends from the lateral wall to uterus to pelvis is the broad ligament. So what are they talking about for suspensory ligament? That's not even in the model. Hang on a second. Ah, here it is. Suspensory ligament projects superlateral to anchor ovary the pelvic floor. Don't even worry about that one. It's not even listed on here. So forget about the suspensory ligament. Just the mesosalpinx, ovarian, uterosacral, and broad. We're not even going to do. They don't even label it.
Can you go in order again with the numbers?
Sure, sure. So let me go back there. So we're going to go top to bottom 14, 15, 10, 1, 12, 7, 13, 3, 4, 5, and then one you can forget, suspensory, 11. Then 8, 6, 9, and 2. Let's see what else we got here. Okay, and here on this page, and we got primordial. Remember, these are primary follicles. These two are secondary follicles. Then mature follicle, secondary oocyte, zona pellucida, corona radiata, first polar body. Like they don't even list that first polar body. Oh, the granulosa cells. These would be granulosa cells around these primary and secondary follicles. And I'm not going to give you a slide. And there's the breast model, nipple, areola, lactiferous duct, lactiferous sinus. These thickened areas right there, really small, but lactiferous sinus. Okay, so that's pretty much it. Like I said, there's more stuff on the female models. So is that everything clear? As clear as I hope I can make it here about the stuff that will. So this quiz, remember, it's a double quiz. It's going to be worth 60 points, not 30, and it's going to be a mixture of. I think that there was one other quiz where I gave you mostly multiple choice, but there's going to be also fill in the blank. We have to come up with it with the structure yourself, and those are going to be worth more points. So that's where I'm starting to design it. 2 points for the multiple choice questions, 4 points for the fill in the blank questions, and there'll probably be more than 15 questions, probably like 20 questions somewhere around there, and it'll be usual thing. You'll have a slide of a female model, a male female model, or ovary model, whatever, and the arrow pointing to something. Either have a choice, select one of the choices, or type in what the correct answer is. So that'll be starting next week. We'll do that next week.
What about for the lab final? How are you doing that one?
Yeah, I'm gonna do it similarly, and I'm not sure the same way as the rest of the online ones. Um, Models, sometimes the actual cat, arrows pointing to different structures, and but there's also going to be regular questions where you short answer questions where you type in the answer, because like on the blood lab, urinalysis labs, those were ones you just typed out in answers. So there's going to be a few questions like that. So I'm going to have to go in to and hand correct. I'm going to have to go in and hand correct the this reproductive models also, reproductive anatomy also, because there's going to be fill in the blanks, and those are machine gradable, but sometimes things have two names, like uterine tube or fallopian tube, and I may put in fallopian and you put in uterine, and it's correct. Or if you misspell something, just one letter off, and of course the computer marks it, gives it 0, even if there's one letter off, it has to be perfect. So, and I don't mark off for for that. So I have to go in and check that, check the computer for the fill in the blanks. So that'll probably be in the last during finals week, I think, is I'll probably get that quiz. I'm just putting together the reproductive anatomy quiz now, and then I'll start to work on that one. It's probably going to be like 25 questions or so, each one probably worth 4 points. It's going to be 100. Could be 100-point exam. So.
And then for the exam 5, I was reading the syllabus. Is exam 5 just the 2 last chapters?
Yes, that is just the 2 reproductive, all of the reproductive chapter, and a little bit on the development chapter. The first few pages of the development chapter that gets down to fetal, just a little bit of fetal development, fertilization, and implantation, and the starting of a pregnancy, and then that's it.
So for exam 5, is it just fill in the blanks or short answers?
It's going to be it's worth a little bit more. It's worth 150, so it's going to be 25 multiple choice questions, each worth 4 points as usual. But then there's going to be short answer questions, or possibly fill in the blank also. That'll be that would be worth a total of 50 points.
And then for the final, like the lecture final, are we still able to do it online and have it 2 attempts? Because I guess the people that can take it with Scantrons in class go to the testing center to take that.
Well, I wasn't going to send anybody to the testing center with Scantrons for the final exam. I made up an online final exam, and I'm going to make it available to you soon. And it's 50 1-point questions, and it's over the whole semester, and it's going to be curved, no matter what. As a matter of fact, I always curve the final exam. The 5th exam won't be curved unless the mean falls below 70, but the final exam is always curved because it's comprehensive.
Is the final open book as well?
Yeah, they're all going to be open book. The only thing that's not open book are the lab quiz. The exams are all going to be open book.
Do we still have 2 attempts on the lecture final?
Oh, no, that's gonna be 1 attempt, but the exam 5 is gonna be 2 attempts.
Okay.
And of course, they have to be taken in proctor track.
Dr.
And
Murphy,
remember, yeah,
for the exam 5, are you still gonna do the second attempt as our final grade, or are you gonna do the median?
oh, yeah, I think I'll probably just take the last attempt as for the
Okay.
grade you get for the, I started that and I think that's better. So I'm gonna
I
do
think so too. Thank you.
that. Okay.
All right, thank you.
I think most students would agree with you. That's a better than the mean of the two.
For
Now,
for the
go ahead.
extra credit exam, it's extra credit. Does the extra credit is there like so many points, or whatever points you get is extra credit?
Yeah, whatever you get on the exam is how many extra credit points you get.
Oh, okay.
Yeah. And the one other thing I'm going to be putting up is group activity 10. That's going to be in a reproductive system, and I need to make that available for you. So that's going to be the last group activity also. 9 is up there now on nutrition, and then 10 on reproduction.
Are we
This-
doing? Oh,
Sorry, if this lab quiz, is this due on Monday or Tuesday? This week? Because last
Oh.
week I think it was different. One was due Monday, one was due on Tuesday.
One, wait a minute.
But
I'm not sure what you
like we had an exam and the lab quiz. Normally they're both due on Mondays, but one week
oh,
they were due Monday.
yeah, I see. Exam, there was an exam due on Monday, and then the lab quiz was due by Tuesday.
Can we do that again?
Yeah, I'll make this available for you guys in section 04. That's purely online. I think I make that available to you a week from today, and then it'll be due the following Tuesday. Now, I'm not sure when I'm going to open up exam 5. I haven't really gotten even started on that yet. But that I can open that up on Monday to Monday. Yeah, I can do that.
Thank you.
for the lab finals and stuff like that, when I was looking at the syllabus, it said that the exam 5 is due on the 14th. Is it gonna be earlier than that?
Oh, what was the question? We're going longer than the 14th. I think that actually the last day of the semester is like the 20th of December, which is really late compared to other semesters.
Oh, okay.
Yeah, so it's going to be due probably past the 14th. That's the last week of the. Let me look at my calendar here.
Because I was thinking finals were like this coming week, and I was like, oh god.
Oh no, they're not. Finals week, I think, is starting on like the week of the 13th of December. I'm pretty sure.
Oh, okay.
And like I said, it ends on Sunday the 20th. So we got we got like 2.5 weeks to go here. The remains of this week and next week, and then the following week is finals week.
Okay. I was just getting nervous because I thought we all had have it all due by the 14th, and I was like, I don't know how we were gonna do that.
Oh no. That's I think I have the connect assignments due, but if somebody needs more time on the connect assignments, I can give you more time. I think I was putting those in the 14th, but I wasn't gonna have everything come together on the same day. But it's tough at the end of the semester. There's a lot of things coming together, right near the end of the semester. So it's real busy, flurry of activity at the end of the semester, and then we finish.
Well, that makes it a lot less stressful when we all know it's not going to be all due on the 14th.
Yeah, it's not gonna be all due on the same day. No. All right.
Are we doing for tomorrow in Zoom? Are we doing activity 9 or 10?
Oh, I haven't even broke open 10. I need to give you copies of 10. I think I'll do that today. Put up copies of 10. But so you can't do 10 yet, but you should be getting 9. If you want to do, some people want to start doing 9 right now. We can I can help you with 9. All right, let's see. Okay,
Can
I'll
we-
stop the recording. You go ahead.
Can we do activity 9 tomorrow in Zoom and then next Wednesday do 10?
Oh yeah, sure. If you want
Okay.
to do that, that's okay with me too.
Okay.
Let me stop the recording here, because I think I got everything for the. I don't want it to be too